guys, so today we're going to talk about the chemistry of life. So this is the first lecture in cell biology. Do not let it scare you. I'm going to try to simplify everything down. Some of it is definitely review. This is one of those lectures that often I'll give and people's eyes sort of glaze over. So depending on your level of chemistry, this could be review. And for some of you, this might be like, wow, what are we talking about? And so that's okay. If you have questions, please reach out and we'll talk about those specifically. So let's get started. Alrighty. So let's get this guy out of my way. And so we're going to talk about the Chapter 2, Chemistry of Life. So this is taken as a mixture of the Albert's textbook and the other text. And so I like to try to make things fun. So, you know, one of the bigger topics in this lecture is free radicals, and they can kill us. Free radicals can be good. We can use them to kill bacteria. But free radicals actually do unfortunate things to our cells as causing signs of aging. But they're normal byproducts of metabolism. And so we'll talk a little bit about metabolism today, but not that much. So here's just some overall key concepts. So just take a peek at these. I recommend as you're studying to look at the study guide, aka questions for Chapter 2, located in your Canvas page. And so let's first begin the difference between a covalent bond and an ionic bond. So covalent bonds, you have electrons that are shared across two pieces of my brain's turned off, two molecules. And in the case of covalent, so those are shared, versus ionic is when one is donated. So for example, like sodium chloride is an ionic reaction. And so in the case of sodium chloride, sodium donates an electron and remains positive. Chloride is negative, and so that is an ionic bond. And so with regard to electronegativity, it is the ability to attract those electrons. And so unequal attraction creates things that are polar. The most common thing that's polar is water. And so here we can see a water molecule. You can see that it is polar. You've got the positive charge over here, positive charge over there, and the negative charge. Versus covalent bonds, where everything is perfectly equally shared all across. This is carbon, the basic unit of life, and it can have 4 bonds. And within those 4 bonds, they are shared equally. And so this covalent reaction creates a really stable structure. So carbon with 4 hydrogens on it's really quite happy. Same token, you can have covalent bonds and create things like double bonds. And so double bonds are very strong. And so here we're showing O2, which is oxygen, and it's the oxygen that we breathe at least. And that is a shared covalent bond, right? And so our biological molecules, such as proteins, phospholipids, they're going to have polar and nonpolar regions. Later when we talk about the plasma membrane, we'll get back into this and we'll get into a term that's called amphipathic, meaning both polar and nonpolar components within the same structure, such as lipids. And so I love to kind of get into like the human perspective. And so first and foremost, we want to talk about those free radicals. I find them so interesting. Possibly it's my age. But these free radicals cause aging, and they're a standard byproduct of our metabolism. And so free radicals are ions. They are often things like superoxide ions. And so they will essentially, if as a free radical, they have this extra unpaired electron, right? So you can see the unpaired electron right there. And so that unpaired electron, if left to its own devices, could interact with other things within our body. So it could interact with DNA, causing DNA damage. It could interact with proteins, causing proteins not to function properly. And so they can become problematic in the sense of we see the younger lady here, and then as we undergo metabolism and as we age, we eventually get to the last stage. I am right here in the middle. So I'm okay with it, but you know, it's good to know. You guys are definitely probably younger than the first person pictured there. And so these free radicals, right, are unstable, right? They're going to have those unpaired electrons, which are trying to bind. They're formed during normal metabolism, so the process of cellular respiration. And free radicals are going to be highly reactive. And as I had said, they're going to impact things like our DNA, macromolecules such as proteins. If they inhibit the action of proteins, proteins function function as enzymes for our cells and do a lot of very important processes. And so if we start to have free radical interaction with our proteins, this is going to lead to additional things like aging, right? And so we're going to start here, and I didn't actually say this on the previous slide, but the word anion, right? That is on the previous slide. And so the learning check is, what is an anion? Does it have a positive charge? Does it contain two or more atoms with a positive charge? Is it a metal and a non-metal? Does it have a covalent bond? Does it have a negative charge, right? So anion versus cation, they are different. So in this case, the anion does have a negative charge, as you can see here, versus the cation will have a positive charge. So cation, my chicken scratch, especially with my tablet kind of up so I can do the camera. And so the formation of free radicals takes place with lots of things, as I've mentioned multiple times, metabolism. But UV light, so especially here in the desert, we've got lots of UV light, and so you can see that UV light causes the formation of superoxide free radicals, potentially OH as well, so hydroxide free radicals. So ionizing radiation, this is a superoxide free radical, O2 with that extra electron, and that can take place because of things like smoking. Nitrogen oxide free radicals also are a product of smoking. Air pollution, so here in the valley when we have the dust bowl that rolls in from LA, that smog or pollution that comes in, that can result in free radicals, so hydroxide free radicals. Our white blood cells, in the case of that first image, they can actually intentionally produce free radicals to get rid of things like bacteria. And cause inflammation within the body. And then, of course, the thing that I'm going to kind of highlight the most, right, besides knowing these other options, is metabolism. So it's a normal cellular process. And so through metabolism, we always form the byproduct of superoxide ions. Those can be converted by an enzyme to hydrogen peroxide. And then we can get hydroxide ions as well. And so we'll talk about some of that cascade. And so just sort of re-illustrating, so free radicals are unstable molecules. So unstable molecules mean that they have an unpaired electron, right? An unpaired electron. And so when they have an unpaired electron, they're looking to be stabilized. They're looking to get in a situation here on the left side where they have stability. They're sharing those electrons or donating those electrons, depending on if it's a covalent shared or ionic donated interaction. And so some of the things that we can do to sort of protect ourselves is the consumption of antioxidants, right? These are a hot topic. And so antioxidants can be in the foods that we eat. For example, blueberries are a really touted antioxidant. And so by having lots of blueberries, pomegranates, coffee, dark chocolate, a little bit of wine, those can help us stop the damage of free radicals. It's all within moderation. And so when we look at this, those antioxidants help us work with our normal enzymes that normally protect us. And so, for example, superoxide dismutase is a very important enzyme during cellular respiration that will convert the superoxide free radical into hydrogen peroxide. But, you know, if any of us have ever seen hydrogen peroxide, like we put it on cuts, you don't drink it, so it's not really safe yet. So there's a next step that we'll talk about here at the bottom. But let's focus first on the superoxide dismutase. And so superoxide dismutase is going to neutralize the superoxide ions into hydrogen peroxide. And they are very important in the sense of it allows for oxygen to be the final acceptor of electrons during cellular respiration. So when we talk about the cellular respiration cycle, the electron transport chain requires lots of oxygen, as does the Krebs cycle. But we're going to get most of our oxygen from the electron transport chain. And so having oxygen present and being able to be the final acceptor of electrons allows us then to get as much energy as possible, or as much ATP, which is cellular energy, for our bodies. And so as a consequence of this metabolic pathway, you have these superoxide free radicals that can leak from the respiratory enzymes, so the cellular respiratory enzymes. So respiratory enzymes, or cellular respiratory enzymes, are proteins, typically proteins. And so in this case, they're converting a substrate into a product. And if we have a buildup of this superoxide free radical, it's going to wreak havoc on the cell. Our cell is not happy when it has things binding in places not intended. So as a consequence, what happens is the superoxide ions can cause mutations in the DNA. It can interact with our enzymes and cause them not to function, or impact other amino acids or essential molecules within our cells. And so as a consequence of that, superoxide dismutase is an enzyme, which is made of protein, that will be able to extend the lifespan of laboratory animals who overproduce it. So they're able to then detoxify superoxygen free radicals more effectively. Catalase comes in next, right? So superoxide dismutase gets us superoxide ions, which then form hydrogen peroxide. So catalase then can take hydrogen peroxide and convert it to water. And so what we have here is we have the O2 superoxide free radical. And that is converted by SOD, right? And then we have hydrogen peroxide formed, right? My chicken scratch over here on the corner. And then from that, we have the enzyme catalase, right? That can give us water, which is perfectly safe for our bodies. And so these enzymes are working together to keep our bodies healthy. And so, for example, in the case of catalase, if we don't have enough of it, it also plays a role in aging and the formation of gray hair, which I have, but my stylist gets rid of it. All right, so other reasons to care about cellular respiration, besides the fact that it gives us energy, when we have a breakdown in things like superoxide dismutase, we can also end up with disease. And so many of you are probably familiar with ALS, amyotrophic lateral sclerosis. And so over here, you can see Stephen Hawking. He is one of the most well-known survivors with ALS. Very brilliant man. And so in the case of ALS, nearly 1/10 of the cases are hereditary, and many of the cases have a mutation in a superoxide dismutase gene. And so superoxide dismutase, as we saw on the previous slide, allows us to detoxify superoxide ions. And so there is some sort of suggestion then that the superoxide ions, all right, are somehow impacting our nerve cells. So here we can see a nerve cell. This is the central cell body. Here's an axon moving away, integrating into a muscle. These are dendrites that are moving into the cell body. And so superoxide dismutase is really important for protecting our neurons. And so leading to the fact that if our neurons get damaged, we can't tell those muscles to function and fire. We have ALS. And so this is a video that I've embedded about metabolism, because I think metabolism is like super scary for lots of people. I'm going to stay on the surface level, but I included this video from the Amoeba Sisters because they break it down really nicely. They'll give you a good refresher. I am not going to test you on the contents of this video, but if you would like a refresher on cellular respiration in like a short chunk, this is a good place to go. So please feel free to watch that. I'm not going to play it in this video so that I don't make my video extra long, but it is available to you. All right, so we're going to begin cellular respiration with glycolysis. And so glycolysis is the breakdown of glucose, all right? The breakdown of glucose and the production of various forms of ATP. And with this, right, so glucose is broken down by enzymes, right? So for example, if we look at the image to the bottom right, glucose would be our substrate, right? And then the enzyme breaks down glucose, and in this case, it breaks down glucose into glucose 6-phosphate. Glucose, I can't even get it to write. Glucose 6-phosphate, right, is actually the product there. And so by being able to break down that glucose, it creates the next product, which is represented by these arrows. The reason I just have arrows is I don't need you to learn these enzymes. I don't need you to learn all the products and all the steps. I just want you to know a few basic things about cellular respiration. In particular, I want you to know how much energy is produced. And so in the process of glycolysis, when we break down one glucose molecule, we get 2 net ATP. And so we get 2 net ATP down here at the bottom, right? And so that's because this process happens twice. And so throughout the process of glycolysis, we insert ATP and we get some ATP out. We insert ATP and we get some out. And so at the end, you get 2. Glycolysis does not take place anywhere special. It's just in the cytoplasm. And so only in the cytoplasm, there's no extra oxygen present. So as a consequence, glycolysis is not oxygen dependent. And then at the outcome of glycolysis, because it goes through twice, right? It goes through twice, you're going to get 2 pyruvate. And those 2 pyruvate are going to feed into the next cycles, which are going to allow us to track this pathway. For the purpose of your exam in cell biology, you only need to know the information that I've posted here, right? Glycolysis gets you 2 ATP. It does not require oxygen. It is located in the cytoplasm, and at the outcome, you get 2 pyruvate, and that just feeds into our cycle so that we know how many ATP we get in the next cycles. And so for that, if you don't have oxygen, right? So if you're at the gym and you're exercising, and you're like breathing hard, but you kind of go past that point where your cells are getting appropriate amounts of oxygen, we go into this cycle called fermentation. And so in the fermentation cycle, we're also going to get a net of 2 ATP. Oxygen is not required, so it's a point where we don't have oxygen. The fermentation video by the Amoeba Sisters does a really good job talking about how you can have alcohol fermentation, which is how we make beer, wine, and of course, breads. And then also flipping back over and talking about the metabolism with regard to our muscle cells. So in both cases, this is taking place in the cytoplasm of the cell that's being discussed. Oxygen is not required. And then in the case of muscle cells, which is really kind of what my interest lies, it's going to create 2-lactate, right? And so when we go to the gym and we have this build up of lactate, the lactate could lead to muscle soreness, right? And so you want to work that out. And part of it is you work to an extreme point where you don't have enough oxygen. There's definitely lots of biochemistry with regard to the muscle lactate conversion process. This is still changing, so I'm giving you really kind of the brief overview. So if you know more or know differently, I'm more than happy to discuss it in class. You probably can teach me some things. But at the end of fermentation, because of the production of NADH, right, we're going to get 2-lactate, but the 2-lactate can be actually converted back up and get us to ATP, which can be fed back through the cycle. And so we kind of like cycle back up and go through glycolysis, getting 2 pyruvate, and so we're back at the stage 1. Now, if we have oxygen, we're going to be able to feed this over, and we're going to go to the citric acid cycle or the Krebs cycle. However you know it, works fine for me. And so there's your fermentation video. Feel free to watch it. I chose some fun stuff. All right, citric acid cycle or Krebs cycle. I grew up learning Krebs cycle. Lots of people see citric acid cycle. I pulled these cool little images from the Khan Academy, so that's another great source if you're going to be taking the MCAT. Feel free to go to that level for studying for your MCAT. You're going to need more details than what I'm covering here. And so at the conclusion of the citric acid cycle, you are going to get 2 ATP, and essentially those are in the scenario of GDP, guanine triphosphate, adenosine triphosphate. And so that's from the conversion of 2-acetyl-CoA, because before we had 2-pyruvate, which are made to 2-acetyl-CoA. The citric acid cycle does require oxygen, and so you must have oxygen, and this is going to take place in the matrix of mitochondria. Now, you see that I have this really cool picture. I didn't talk about any of it. That's because I don't need you to memorize anything else on this slide except for the fact there's 2 ATP/GDP. Oxygen is required, and this takes place in the matrix of mitochondria. So for the electron transport chain, this is really where we get all of our energy. And it's because, yet again, we have oxygen, and we're now moving from the matrix of the mitochondria to the inner mitochondrial membrane. And so all of these hydrogens, right, there's some cool like YouTube videos, and they call these hot potato hydrogens. All these hydrogens end up feeding through this gradient and going through an ATP synthase. And so the ATP synthase is going to produce ATP. And depending on how effective that process is and what sorts of cofactors take place here on the left, you may get lots of ATP, as many as 38, but at least 36 ATP from the entire combination of all the cellular cycles. So 2 from glycolysis, 2 from the Krebs cycle, and then, right, about 32 to 34, right, from the cycle here. All right, so let's do a learning check. So first statement is the chemistry of life is carried out and coordinated by primarily the action of small molecules. And so this is a time that it's helpful to define what is a small molecule. So small molecules are things like oxygen, right? Sodium would be a small molecule, right? And so chemistry of life is kind of like cellular respiration. And so is cellular respiration solely called out by oxygen? Or are there enzymes, which are protein, that are involved? So take a moment to think about that. I'm going to pause and try to give you a moment to think. And so are proteins small molecules? Because they are pretty darn important for cellular respiration. All right. So hopefully on this one, you decided that this is false. And it's false because the small molecules thing, right? So proteins are not small molecules. Proteins are actually fairly complex and fairly large in size. And proteins are required for the processes of metabolism. And so they convert all those substrates into new products so that we can go through those cycles. All right, part B. Carbon-based compounds make up the vast majority of molecules found in cells. Carbon-based molecules. So what are we mostly? Are we carbon? Are we nitrogen? Are we oxygen? And so this answer is true. We are predominantly carbon-based molecules. The fact that carbon can have 4 bonds makes it really important and creates a fabulous building block for life. So part C. The chemical reactions in the living system are loosely regulated. And so I'm going to underline loosely regulated, allowing for a wide range of products. You know, anything can come out and more rapid evolution. So this is definitely false, right? And key terms are the fact that it's loosely regulated. So cellular respiration has very specific enzymes at very specific parts. And we know what product come out, right? We don't have random products. We're just like, oh, it's not like a grab bag of like, what's next? We do still have evolution because of things like superoxide free radicals causing mutations within our DNA. And anything that's beneficial we keep. But, you know, it's not taking place loosely. It's very orchestrated. All right. So let's switch base. We're going to talk about acids, bases, and buffers. And so acids are going to donate protons, right? Bases will accept protons. And so that makes them unique. So here we have acids, right? And this is a hydronium molecule. Over here we have a base that is a hydroxide molecule. And so in the middle, right, we get to neutral pH, which is 7, right? And the acid scale, we can go as low as 0. Most things are like something between 1 and 6. My chicken scratch. My other program writes better. And then on the case of bases, these are very alkaline. And so alkaline components are anywhere between 8 and 14. And so if we look at this scale, we're really looking at the quantity of hydronium or the quantity of hydroxide. And so, for example, right, at a very low acidic pH, right, you can see that you have a tenfold like pH. Whoops, sorry. And so at a pH of 1, you have, oh, I keep getting close to this edge. It's not, oops, it's not liking that. No, it's not letting me zoom in on this. And so at a pH of 1, we have 10 to the negative 1 hydronium ions and 10 to the negative 13 hydroxide ions. It's kind of how that works. They're sort of inverse in relationship. And so keep in mind whether something, if it's acidic, there's often we think of it as having more free hydrogens, right? So hydronium can donate a hydrogen and become water, versus OH is negatively charged. And so OH, to be able to become neutral, it needs to accept a hydrogen ion to become water. And so hydroxide ions are very reactive and alkaline in pH. Hydronium ions are very acidic and can donate those hydrogens to become stable as water. And so these are all balanced out by buffers. And so our blood, for example, is buffered by carbonate, right? And so depending on cellular respiration or our oxygen flow, right? So carbon dioxide, if we get a lot of carbon dioxide in our blood, we can start to form carbonic acid. And so carbonic acid causes, actually, the carbon dioxide causes us to signal respiration. So we start breathing more, right, to balance that back out and reduce the pH of the blood, because we need the pH of the blood to stay really, really stable, because it's going to impact the function of proteins. So acids and bases will actually alter the function of proteins. We'll talk more about that in chapter 4. And so here at the bottom, you can see very weak acids versus very strong acids, right? So things like hydrochloric acid is a very strong acid. Things like hydroxide is a very strong base, right? And then in the middle, you have things like water. And then on the far right, when we look at this diagram, right, so you can see you have a polar covalent bond right there, meaning it's very reactive. And so in the presence of water, the acetic acid can be broken down into, converted into acetate and hydronium ions, right? So what is the pH of pure water? Hopefully you picked that up in the previous little couple slides. So the pH of water is 7, all right? And the concentration of hydroxide or hydronium at a pH of 8, which is greater? It's a pH of 8, and so pH of 8 means it's going to be basic or alkaline. And so if it's alkaline, do you have more hydronium, which is H3O, right? Or hydroxide, which is OH negative, right? So OH negative, H3O positive, right? And hopefully you said hydroxide. All right. So we're moving right along. We're going to talk about these small molecules becoming much larger molecules, such as sugars becoming polysaccharides and complex carbohydrates. And so carbon is central to the chemistry of life. You know, the fact that there are 4 covalent bonds with itself or other atoms really makes it this dynamic structure. And so when you get to organic chemistry, you'll really just learn to love all things carbon. And so carbon-containing molecules are called biomolecules. And so as we have these small building blocks, which is what we're going to talk about today, we'll talk about sugars or carbohydrates and how those sugars or carbohydrates can form complex chains of polysaccharides, so meaning many sugars. And so carbohydrates, lipids, proteins, and nucleic acids, these are the 4 types of macromolecules. right? And so these are really critical to the function of the cell, and they have really important roles. So for example, in the cell wall, we're going to talk about carbohydrates a lot. Those sugars make a darn big difference. For example, our blood types are determined by sugars. Likewise, when we think about proteins, proteins are made of things like microtubules. So microtubules form spindle fibers. Hopefully you remember that from BIOL 196 when cells go through cell division. And so spindle fibers made of microtubules, which are protein, are really important. If you don't have them, the cells are not going to split in two. Likewise, pieces of the mitochondria have proteins or transport proteins such as ATP synthase. ACE is often the word of an enzyme. And so ATP synthase in the mitochondria is how we get so much ATP from the electron transport chain. And then when we look at the ribosome, so the ribosome is made of two components. It has a protein level component, but then it also has RNA involved. And so very unique structure, and we'll talk about that a little further. And then the plasma membrane is comprised of lipids. And so lipids I find so interesting. And so we'll be talking about lipids. And then, of course, we're going to end off the lecture today talking about different types of nucleic acids. And so things like our DNA and our RNA are nucleic acids. And they work really tightly with things like proteins to keep us healthy. All right. So I took a little pause there because I'm like, wow, I've been talking for a long time. So we're actually going to stop with this lecture for today. We will pick up with part 2 of chapter 2 for you to watch. So I'll be back.